right, so let's get started with this next session of the cell size and growth seminar, and we'll get started with Benedikt Mairhormann from the Schmoller Lab in Munich. So, Benedikt, if you can share your screen, we'll get started.
Yeah. Here we go. Can you see my slides?
Perfect.
All right. So thanks, Jan, for the introduction. And thanks for the opportunity to present my PhD work here in the seminar. So I'm doing my PhD, just wrapping up in the labs of Pascal Feiter-Braun and Kurt Schmoller. Most of you probably know Kurt. He was also postdoc with Jan. And what we're doing in this lab is trying to understand how cells regulate their size, which probably is also very common in this crowd. And if you think about it, the way cells manage to regulate their size is by regulating growth against division in the cell cycle. So what we also found at the beginning of my PhD, or what was initial motivation, was that most of what we know about this is found by looking at several lab strains like W303 or S288Z, but we don't really branch out to the species of S. cerevisiae. And actually, there's lots of variation which we don't really cover in the lab strains and which was uncovered in recent studies like 2018 in a study where 1,000 yeasts were isolated all around the world and sequenced, and kind of this whole phylogenetic tree on a species level could be constructed. And this is a very interesting resource and very promising resource for us, which we wanted to look into. What we do in Kurt's lab mainly is microscopy, and the first question, of course, we asked was, is there phenotypic variety within those 1,000 strains? So we looked at a few, and those images are just a bit foreshadowing. There is definitely lots of phenotypic variety when it comes to cell cycle progression within those strains. And the second very interesting question, given that we also have other resources on those 1,000 strains, like a whole data set for RNA-seq and another data set for mass spec, is if we can understand which variation in those regulatory layers actually has an effect on cell cycle phenotype. And we're hoping that we can learn from this variation and what of this variation is causing cell cycle phenotype. I mentioned we work with microscopy a lot, and one very central bottleneck which would prevent this kind of a study is at the image analysis. So if you want to study the cell cycle, we have to look at those visual markers for transitioning from T1 to S and at the division, and this means, of course, we need videos. And if you do time-lapse microscopy, and if you analyze time-lapse microscopy before, then you will have realized that this is a very tedious process with lots of clicking back and forth in between frames in order to segment the cells and track them, as you can see in this video. But also, which is very important to us, in order to annotate when the cells are transitioning through the cell cycle stages. So we want to pair mother and daughter cells, and as soon as they divide, we also want to have this precise moment of division annotated in our videos. There was no real good end-to-end method, and that's why I was hired in the first place to develop such a method. My background is more in mathematics and machine learning, deep learning. So I trained neural network which uses the time context, so kind of mimicking the human behavior when we do this manually. And I trained this neural network on data which we generated with our in-house tool cell XCDC. The model is trained to predict 6 channels, which are pixel level features. So first of all, the model tries to predict for each pixel in which cell cycle stage the cell is, which this pixel belongs to. This is G1, SG2M as a daughter cell, and SG2M as a mother cell, abbreviated like you see here. And then the second set of features are those morphological features. Here, most importantly, we have this bottleneck, which we can construct from the annotations we have in cell AC/DC. And this is then also predicted by the model. So model tries to already predict pixel features to later on pair mother and daughter cells. And this is how this looks in time. As I said, the model also sees temporal context. So it's trained on patches of 8 frames with some XY context, and then predicts those channel maps. But this doesn't really solve our problem, right? Because now we only have nice looking videos, but we want to actually go into cell cycle analysis. So the method is not yet complete. We need to take those pixel maps and now in post-processing transform them to actually solve the tasks that I was mentioning earlier. So cell detection is done by some post-processing. Then for tracking, I integrated an existing method, which is Tracastra, which works very well, as I found. And then the most important part, the cell cycle annotation, where I extract daughter SG2M phases and try to map them on the mother SG2M phases with some optimization routine. And of course, if you develop a method like this, you need to benchmark it against the state-of-the-art methods, which I did. And I don't want to go too much into detail. This is kind of the overview figure where I have all the metrics or many of the metrics for segmentation, tracking, and cell cycle. You have to believe me that my method or the method that I developed over the years really works better than state-of-the-art methods. And that is even if you take the state-of-the-art methods like CellPose or Yeezy and retrain them on our data set, which is way larger than, or which is a very, very large yeast data set with the annotations from cell AC/DC. So we have a method which works well, and this is then kind of what the final output looks like. Sorry. So I built here the predicted pedigree on the right, and this is just for the left subpopulation, just so you know. And you can basically see that what we get earlier or what we got earlier from manual annotations, we now get in a fully automated fashion from the method, and what we get as a final product, basically, or final result, are those pedigrees where we have full lineage of the population that we see in the video. And this was kind of the first step, which I worked on for quite some time. Now, moving on, we wanted to actually discover biological insights with this. So I went to the lab quite a lot. In our lab, we have this, or we use a custom microfluidic setup, which is developed by Igor Kuchtevich, who is also an IFE in our institute. And it's very cool. You can basically inject and image 8 strains at the same time in those 8 separated subchambers. This is the data you get. And then with this setup, I, as I said, did lots of experiments in the end, ending up with, I think by now it's more than 1,500 videos. And we have at least 9, I would say even a bit more videos per strain from 2 biological replicates. And with this, we went through 98 strains plus 1 reference strain, which in my case is S288C. So if we take a closer look at those pedigrees, what do we want to do with those now? If we look at one example cell here, cell 29, switch this on, you can see that basically this pedigree data is very rich in information. First of all, we have an SG2M phase where itself, the cell itself was a bud at the beginning and growing, still connected to the mother cell. Then we have cell division. We have 2 G1 phases, one of the daughter, one of the mother cell. And then we have this next SG2M entry, where the previous daughter cell now becomes a mother cell itself. And this kind of we laid out, and then from this first 1.5 cell cycle, we extract several features, several phenotypes, like the volumes and the growth at the transition points. So at G1S entry and at G1S transition, and later on at division, and in between, basically, then also the timings, how long those cell cycle stages are, and then a set of more specific features, which is size control and asymmetry. So measuring how asymmetric in volume or in cell size terms those cells grow and divide. And this leaves us now with 11 phenotypes, which describe cell cycles. And if you think about this as a data set, the special trait of this data set is that the cell cycles are aligned. So we compare volumes at exactly the same time, not in absolute terms, but in relative time within the cell cycle. And 11-dimensional data, it's kind of hard to approach. So I did some dimensionality reduction and visualized this with UMAP here, which hopefully most of you know. This is basically on first sight showing that the variation that we see on the single cell level is not really strictly separated clusters, but it's more like a continuous space, which probably would also be what we expect. And then just to drop some numbers again, this is 9.2 million observations of single cells at a time now, amounting to more than 100,000 cell cycles. So it's quite a large data set. Now we can go ahead and do some more explorative analysis on this data set. So for example, I projected 2 of the phenotypes onto the UMAP, and we can see that we have this area on top here, which is dominated by high values in elongation, so more producing elongated cells. And then this area on the left is mother volume at budding, so it's just generally large cells. If we now go further and look at some strains, we find some strains which are concentrated quite a lot within those regions in the UMAP, like this strain, which probably is quite elongated cells, this, which is large cells, and then this I put as reference. This is the reference strain, actually. And then you can also go ahead and look at single cell cycles, and you will see exactly what I just talked about, that basically we have very elongated cells from the one strain, very large cells with short SG2M phases for the second strain. And this cell cycle is actually quite funny. It's the most boring cell cycle, which is selected by lowest set score overall. So this is the most standard cell cycle out of the 100,000. Now, this is still very hard to work with data because it's single cell, you have different numbers of samples per strain and so on. So the next step was to go to the strain level with a very simple aggregation. So if you look at, you can see one example phenotype, G1 growth, for 3 different strains. And the data points here are single cell cycles, and I undersampled a bit the reference strain because there's actually massive numbers. You can calculate the average of those cell cycles. You get the mean, and you can calculate the coefficient of variation. So for each strain, we can extract 2 quantities from the distribution of the single cell cycles to get a strain level representation. So here on the right side now, every data point is 1 strain. And the phenotypes we're now looking at is the mean of the G1 growth of the strain and the coefficient of variation of the strain of this one feature. Just to give you an example, of course, we do this for all the features, which leaves us with 22 phenotypes. So now we have the 11 single cell phenotypes, and by strain aggregation, we get double this number, taking mean and coefficient of variation of each. And this data is now better to work with the available data that we have, with the RNA-seq data, with the mass spec data set, and also with the data from DNA sequencing. So I, again, because this is high dimensional data, did some dimensionality reduction, this time linear with PCA. So here you can see each data point representing one strain. What we did next was to detect outliers. So to detect strains which show an anormal cell cycle progression with a statistical method. And this gave us those 23 strains in total, which can be considered as outlier strains. I then also did a clustering on those outlier strains, and I found two very nicely separated phenotypic signatures on the strain level. So this is cluster one and cluster two, two clusters of strains which show non-reference like cell cycle. And if we look at the features at the phenotypes very quickly again, we can see that this is also represented there. So if we look at one feature or one phenotype, then basically typically one cluster will have a bit higher values, the other will have a bit lower values. Or as we can see here, one cluster is very reference-like, the other cluster has some deviation in this phenotype. And now we took those labels. So we have strain data now. We have those strain level labels for the strains. And we went ahead and went into actual biological discovery by asking the question if we can predict cell cycle outliers, and if yes, which modality do we have to look in? So we started with some omics data, like genomic variants, RNA profiles, or protein profiles. Here you can see a heat map of the protein profiles, or another example, genomic variants, which are binary data, so presence or absence of a genomic variant. Then we implemented a very comprehensive machine learning pipeline where you would take all the combinations of those input data sets, or you take all the input data sets and predict systematically, is this an outlier strain? Is this cluster one or cluster two? And then the plan is to take the best models, if we get some that work well, and analyze the feature importance. So we get this gene level scores of how important the genes are to predict if a strain is going to have an outlier like cell cycle or not. So did this work? The answer is definitely yes. We could find lots of scenarios where we were surprised of how well you can discriminate between cell cycle outliers and no outliers. Generally, I think one insight was that protein is definitely the most informative data source for this task. And the best models were models based on protein data. What we now did next was to pick the best models, and it was really like a lot of scenarios. So I'm now following up on two scenarios in particular where the models were just almost perfect. And then looking at in this case, it was also logistic regression models, so very simple models, very explainable models, and then listing or sorting the genes by their contribution to the prediction. And then we get this list of features which are important, or genes which are important, like TES1, or which are negative, which are also important, but in a negative way, like ATP5. And the next question now was to reduce those models to predict cell cycle outliers from as least genes as possible. And that's what we did. So basically, I took the top 100 and then threw out the 50 genes, then threw out another 25, and I went down, down, down. And interestingly, we can actually get almost perfect predictions for this cluster 2 outliers from just two genes. And this is not only INH1 and PAF3, which I'm showing here, but this is several combinations of two genes which allow us to make almost perfect predictions of if a strain is going to be a cell cycle outlier or not. So this brings me already to the outlook and to the summary. what we want to do next is we want to do hemizygous deletions. So we have homozygous diploids. We can just delete one copy of genes. And then our hope is that we can rescue the cell cycle phenotype in the outlier strains by reducing the gene expression in the positive predictor gene, or induce the cell cycle phenotype in strains which right now are no outliers, but maybe by knocking or by deleting the gene, we can induce this phenotype. So yeah, let me summarize once more. We developed an end-to-end method to automate this process to get pedigrees out of videos. With this, we generated a quite large data set of 100,000 cell cycles of 100 or roughly 100 different strains. And now we try to systematically analyze this and by predicting outliers from omics data. And our hope is that those few models that we found, which work very well, are actually not only uncovering perfect correlations, but there's actually some causality behind this, and we can now reproduce this effect by deleting single copies of the gene. With this, thank you very much for your attention and looking forward to your questions.
All right. Great talk, Benedikt. So, all right. As usual, please put your questions in the chat as you have them, and we'll go through them in turn. So while people are doing that, I'll start with the question. When you did the logistic regression there at the end to determine the contributions of the different genes, I noticed that the parameters or the coefficients rather for each one were pretty similar quantitatively. So does that mean there's like a lot of kind of redundancy in the prediction? Like, are they producing kind of similar contributions? In the end, you pick out two here and then create the hemizygous diploid. But from the previous analysis, it's not clear to me how you get to those two if so many genes have really similar coefficients. It's not like there's just one that jumps out, right? Or
Yeah.
do I misread the?
Yeah. So you mean this one, right?
Yeah, that
Yeah.
one.
So so this one is like those feature importances are from a model which takes in 500 genes as input. And then when you analyze the feature importance, it's a bit blurry, as you say. So they are very similar, those values. But then if you go ahead in the next step that I mentioned, that I like iteratively reduce the number of genes that the model has available, then you get more informative feature importance values. And also, in the end, when you arrive at two genes, then it's just those two genes are definitely important, right? So this is just the starting point, basically, just the first ordering of the genes, and then we go down that list and basically eliminate genes on the way, and then we get effective feature importance by looking at models which only use two genes.
Okay, I see. So it's just part of the like kind of truncation of the model.
Yes. So
Right.
it's always new models, but the new models, they use less and less features.
Okay, great. All right, so we got a bunch of questions here. Dave, if you want to unmute and ask your question, which I think will also help, I think, clarify for some people how this modeling was done.
So, beautiful talk. Are you saying, well, let me just get to the conclusion. By focusing on pairs of proteins, pairs of genes, are you saying that all the proteins that you cone down to actually interact with each other? It doesn't matter which two you pick because the whole set sort of interacts with each other?
No, I think that's maybe one option that all of those genes that or many of those genes that end up in those two gene-based models are actually taking part in regulating the phenotype. But I think it will be more like we need to start somewhere now. So we pick combinations of genes that just work well, and we do our homozygous deletions, but one would probably need to systematically test through all of those genes to really understand which genes in combination with which other genes are causal for the phenotype. So the answer is we don't really know yet, but we're starting to go in that direction of testing this.
But isn't there already a database of interacting proteins?
So you mean physical protein-protein interactions, or?
Yes.
Yes. Generally, looking at our candidate genes, we didn't really find lots of prior evidence of anything. So one thing that was remarkable was that in one list of candidate genes, there were lots of mitochondria-related genes, which is especially cool for Kurt because that's also the direction that we want to go in the future. But those genes, like there's nothing known about physical interactions in between the genes that I found.
Thank you.
Thanks.
All right, next up we have Cesar. Cesar, if you want to unmute and
Hello.
ask your question.
Hey. Hey, how are you? Nice talk. Yes, so I'm working currently in different kind of cells, work with bacteria, fission yeast, mammal cells. I was wondering if how difficult it is to implement this kind of algorithm or to make a general platform that can like work with these different kind of cells
Yeah.
using
So
similar methods.
I think the answer is a bit divided. So starting from the image analysis, this is quite specialized on budding yeast, but one could take the same model and construct those input features. Maybe I can quickly go there at the very start. Sorry for that. So one could go and think about the analog of those channels in the respective cell type, right? So if you have fission yeast, it might be quite similar actually, and maybe you could think of the butt neck in fission yeast also as this like ring which is contracting. So you could construct something similar, and then what you definitely need is training data to get the method started, and then I could imagine that something like this works well in other cell types as well. When like the second part for the machine learning and for this explorative analysis, that's quite agnostic. So this, in principle, you could use with any omics data on the one side and phenotypic data on the other side.
Great, thank
All
you.
right. Great. So next up we have Jamie. Jamie, if you want to unmute.
Great. Yeah, thanks. So really nice talk, and I love the use of these wild strains. I was wondering if I just didn't know if it's known if S phase onset it happens at the same time as bud emergence in all these wild strains, or like do some of them have differences in when, say, G1S versus G2M might be happening? Is that known?
Yeah, it's actually a very cool question. So when I talk about SG2M phase in this like image analysis context, I talk about first detection of the bud. What's really going on within the cells, I think that there would be a cool future direction to look into for some selected strains. I think when you would integrate some fluorescence markers. Cool thing about the method, like about the image analysis method, is that it works without any channels. So it just works from phase contrast. So we deliberately kept all the fluorescence channels free. So I guess would be an interesting next step to look into that.
Yeah, cool. Thank you.
Thanks.
Great. So next up we've got David.
Thanks for the talk. The technology looks amazing. We work in aging yeast. And what we see is that when the yeast ages, the size of the mother increases. And at some point, some of them change their morphology to the elongated type. So I was wondering if you think that maybe that could show up in the UMAP somehow, like, I don't know, transitions from one extreme to the other in the UMAP. Have you thought maybe about that?
Yeah, so yeah, it's a very cool question. I think aging in general, we are a bit limited because in our setup, the field of view is just full at some point, right? So the deepest lineage that we get is maybe 7 or 8 generations. But in general, so I took those strain density maps here. I took them from 100, right? So the question that you're asking would be like, are there strains which have lots of cell cycles in this elongation region, but also have cell cycles somewhere more central? And yeah, we can definitely look into it, and then you could go and then reanalyze or like more specifically analyze the data from the strain and see if there's a correlation with the replicative age, that the elongation only appears after some divisions. Yeah. So yeah, in principle, you could look into that. I didn't really do it yet, but it's a nice idea.
Cool. Thank you.
All right. And finally, we've got a question from Ivan.
Very nice talk. I have a practical question, like similar to Cesar question. Does your framework require training for datasets acquired in different conditions, let's say in a different microscope with different objective, and kind of like expansion of this? Would you expect this approach in general work for bright field images?
Yeah. So yeah, it's also very nice questions. Thank you. So I haven't really spent any time in making this a generalist method. So for me, it counts how well it works in my setup, and that's it. But I think like so answer to the first question, I think definitely retraining would be needed. And we also when we publish this, so I'm just working on the preprint. When we publish this, we will also publish code to retrain the method and yeah to get this method like going in multiple setups. For the second, what was the second question again? Sorry. The bright field. Sorry, now I get it. Yeah. Bright field, we have very concrete plans, and the idea is that we do this kind of transfer from phase contrast to bright field by using predictions of the model. So taking data from phase contrast and bright field together, then predicting on the phase contrast, and for a first step, use the predictions as ground truth for the bright field. So that's the plan. I yeah might even work quite well. And then probably there will also be at least some manually curated data sets we will need to train on bright field as well. But we want to also do this on bright field. Yeah.
Okay, I see we just got one more question, but we're gonna move that question over to the Slack channel so we can get started with our second speaker. The other thing I guess you could say is like it's not that expensive to buy a phase objective. So if you have a method that's already really working, it's probably much cheaper to buy a phase objective than to do all the work to retrain, generate the ground truth data in a different system and retrain it. Okay, so sorry, Drish, we're gonna have to move your question to the Slack. And Yiqiao, if I don't know if I'm got the pronunciation at least vaguely right. If you could share your slides, and we'll get started with the second talk in the session, which seems to have a theme of strange yeast cell cycles. So I think Yiqiao, are you there?
Yes. I am having difficulty just sharing my. Oh, yeah. There we go.
You should be able to.
Yes.
Yes.
Are you only seeing my PowerPoint, and is it also the whole screen?
We see your well, like the whole PowerPoint. So start the show and let's see what happens. I feel like it's gonna work.
Yes.
Feeling good.
Oops.
Looking good.
Okay.
Great.
I can't see you guys. Oh, okay. I can. Okay. So, yes. Do I have a little introduction, or should I just go ahead?
No. The introduction is now we have Yiqiao, and she's going to tell us about this multi-budding yeast. All yours.
Thank you. So today, actually, most of the contents I'm going to talk about today are work in progress and a lot of puzzles. So I probably don't have answers to many of your questions, but I still appreciate your inputs and thoughts or just crazy ideas. So today I'm actually going to tell you about looking at cell size control, but through the lens of cell cycle regulation in this new fungal model that the Love Lab have been working on in the past 5 years that is called Aureole Basidium prurulens. To start with, I actually wanted to bring everyone to this background where it has observed for a long time that in two model yeast system as Cerevisiae and Pombe, the cells have a typical size and shape, and the distribution of the cell size is quite tight. For example, Cerevisiae haploid cells are typically 4 micrometer in diameter, and then diploid is 6 micrometer. And then Pombe enters mitosis around 14 micrometer at a abundant nutrient conditions. So this regulation, or this very tight size distribution, is in part due to the type coordination between cell cycle progression and cell size. So at the simplest level, omitting a lot of details, in Cerevisiae, the model is more like when a cell was born smaller, it will spend longer time in G1 to reach some size threshold and then to enter the next cell cycle. And then in Pombe, the regulation is more at the G2M transition, where a cell that were born smaller, it will spend longer time in G2 to elongate further before it can go through the G2M transition. Now, at the molecular level, the coordination of cell cycle and morphogenesis is mainly carried out through the activity of cycling CDK regulating both the cell cycle and morphogenesis. So here I'm using Cerevisiae as Saccharomyces cerevisiae as an example, where G1 CDK activity regulates both G1S transition and then bud emergence. On the other hand, the mitotic CDK activity regulates depolarization and then transition between from G2 to mitosis. Now going further into detail, there are two major, I wouldn't say checkpoint, but then regulatory points in the cell cycle where the cell will actually check for its morphology. So one is at this G1S transition, the cell will ask that, am I big enough? So it monitors its size. So only when the cell reaches a size threshold, it then activates, or the G1 CDK become active, that triggers DNA replication and budding. So another point is a G2M transition. The cell will monitor whether it has made a bud, and only when a bud has been made, mitotic CDK activity would then become active and then triggers entry into mitosis. So a lot of molecular detail has been worked out in Saccharomyces cerevisiae in a lot of labs. But then the Love Lab started working on this interesting new fungal system that does not seem to inherit any one of those two principles. So those are the things I'm going to tell you about today. So Aureole Basidium prurulens, it has actually several ways of proliferation, but one way is through budding. So here I'm showing you examples of cells with either a single nucleus or multiple nuclei. So the histone protein are tagged by an cherry tag, and then it's pseudo-colored to magenta. So I hope you can appreciate that in a single nuclear scenario, is looking actually very similar to Saccharomyces cerevisiae. And then in the melted nuclei scenario, the nuclei actually go through the cell cycle quite synchronously. And then at the end of the cell cycle, the bud can inherit one or two nuclei. So previous quantification by another postdoc in the lab have found that a single plurulence mother cell can contain variable number of nuclei, make variable number of buds, and then can have a huge variability or differences in size. Because the interest of, so my interest is looking at, is there any coordination between morphogenesis and the cell cycle? But to start with, we really have to first understand how does the cell cycle look like in plurulence. So to do that, I actually developed two fluorescence probes. One is tagging PCNA, is a component of the DNA replication machinery, which will mark the S phase, which will be very bright puntas. Here will be like black. Those puntas marks where S phase is happening. So before S phase, between the previous mitosis to S phase, I call G1, and then after S phase to the current mitosis of the current cell cycle, I call G2. So one feature from the multinuclei cell is that you can see the nuclei are actually going through the cell cycle quite synchronously. Now, by going through a lot of these live cell images, I was able to then summarize what the cell cycle looked like in plurulence budding yeast cells. So different from Saccharomyces cerevisiae, which it has a much longer G1 and a negligible G2, plurulence cells actually have a pretty short G1 phase, but then a very long G2 phase. If we then look at individual cell and then look at what is the distribution of cell cycle phases in the population of cell, we could actually see that there's also a huge variability. For example, if you look at the histogram on the right-hand side, there are cells only spend maybe like 30 minutes in G2, but then there are cells that spend up to 2 hours or even more in G2 phase. So that brings us to the first question, like where does this variability come from? So a very obvious prediction or model that comes from what we know is probably variability in size. So for example, in Saccharomyces cerevisiae, adopted cell born to a mother are always smaller. So then because the cells that were born smaller will spend longer time in G1, so that's where the variability in G1 length can come from. However, when we split the plurulence budding yeast cells into mother cells on the top row or buds on the bottom row, and then ask what is the relationship between the cell cycle duration, different phases, and then the size of the cell at birth. We actually did not see a strong correlation between these two parameters. So it seems that the cell cycle phase duration is not directly correlated with cell size. So that is a correlative analysis. So the other thing we could do is to ask what about those molecules that we know to regulate or be important to regulate the size threshold in cerevisiae. So that brings us to the V5 gene. So in Saccharomyces cerevisiae, at the simplest level, V5 inhibits cycling, specifically CLIN1 and 2 expression to inhibit or delay G1S transition until the cell have reached a size threshold. Now, when we delete V5 in plurulence, what you can appreciate is actually the size of the cells are actually pretty similar to wild type, even look at a very large population level. And then we also did not see any differences in the cell cycle profile. So the V5 the list cells also have a short G2 and then a long, short G1, long G2 profile. So it seems that V5 by itself is not the answer, or V5 is probably not playing a role in if there is a size control in Poulence. So this is the summary of the first part. In Cervise, there's a size control point in G1 as late G1, where only upon obtaining a certain size, the G1 CDK complex will become active and then triggers DNA replication and budding. But it does not seem to be the case in Poulence. So in the first part, I talk about a active CDK cycling complex that is required to trigger G1S transition. But there's another point in cell cycle where at G2M transition, the V1 kinase actively phosphorylate the active CDK cycling complex to inactivate it until certain condition is achieved. So in Saccharomyces cerevisiae, when deleting the V1 homolog, it's called V1, in like happily growing cells, there wasn't a significant phenotype in the cell cycle or the size of the cells. In POMB, however, when V1 gene was deleted, the cell will immediately have a much shorter G2. And in the cell, because they were actually entering mitosis much earlier, so the cells are shorter. And because of that, the subsequent G1 cell cycle phase is actually lengthened. So phenotypically, this is what it looks like when V1 gene is deleted in POMB cells. So wild type cell have a 14 micrometer length when at the G2N transition, but then the V1 deleted cell are much stubbier and shorter. So what happens to Poulence if we delete the V1 gene? So to our, it's more like relief, when we delete V1, we actually also saw a shortening of the G2 phase and then lengthening of the G1 phase. And then the total doubling time also becomes longer. But then the next question is, why is this lengthening of G1 cell cycle phase? Is it also because the cells were once smaller? So here is where the first surprise came. When we quantify the size of the bud right at cytokinesis in wild type and V1 delete cells, we saw that the V1 delete buds are actually not smaller, maybe slightly, but then in general, they are pretty similar to the wild type cells. So why are the V1 delete cells not smaller? So we then need to go back to when in the cell cycle does a Poulence cell make a bud, and when does this stop? So now going back, so the top row is the wild type cells. So in wild type cells, bud emergence happens in late S phase and early G2 phase. So the cell will have this entire G2 or maybe even longer to make a bud. So if this is also the case in V1 delete cells, so because now it has a much shorter G2, it will essentially make a much smaller bud. So it turns out when we look at bud emergence in V1 delete cells, the bud emergence actually shift entirely to earlier in the cell cycle. So on the movie on the left, actually this is an example where a V1 delete cell starts a bud in G1 phase of the cell cycle. So it seems that one explanation of why the V1 delete cells, their buds are not smaller than the wild type is because they make it possible that they initiate the bud much earlier in the cell cycle, so then it have essentially a much longer time to grow the bud. So besides the changes in cell cycle duration and then the budding timing, another interesting phenotype in the V1 delete cell is a significant increase in the percentage of cells that go through mitosis without even making a bud. So this is another interesting way that purulent cells can that proliferate. So they can go through non-bud cell cycle, that's what we call them. So they basically grow isotropically, double the number of nuclei at the end of the cell cycle, but then without even make a bud. So this is a little summary of the second part. So in wild-type cells, the cell cycle looks like a short G1, long G2, and a bud emergence happen at late S phase and early G2. When we delete the V1 gene, now the G1 cell cycle phase is longer, G2 is shorter, and it does not seem to be, or the lengthening of G1 is not due to changes in the cell, the bud size at the end of the mitosis. So now if we think back to the first part where we did not see any phenotype in the V5 single delete mutants, what if V5 is still doing its thing well, maybe its function is still regulated something related to G1, but because in wild type, the G1 is so short that probably the V5 function is not important. So actually just out of my personal curiosity, I make this string where I delete the V5 gene in the V1 delete background that has a much longer G1. So is it possible that when given longer time, the V5 function will be more important? So here is the comparison of the budding behavior in wild-type cells versus the V1, V5 double delete. So the PCN1, which marks replication or S phase, is in cyan, histone is in magenta. So after when I quantified the cell cycle duration, what I found was that V1 delete or V1, V5 double delete both have a longer G1 and then a shorter G2, but then there's no significant difference between these two strains. So at least this tells us that V5 is not the explanation of a longer G1 cell cycle phase in the V1 delete background. So I don't know whether you would notice another interesting feature of the V1 delete, V5 double delete, the double delete mutant is that each cell contains seemingly more nuclei. So that's what I then ask. If I calculate the area, the ratio of the area of the nucleus and then the area of the cell, what I found was that the V1, V5 double delete cells have a much higher nuclear to cell area ratio. But that ratio does not seem to affect actually the cell cycle in this double delete mutants. So the temporary conclusion we can draw now is in a V1, V5 double delete strain, the cell cycle actually looks pretty much similar or identical to a V1 single delete cells, but then per area of the cell, there are way more nuclei. So what I think is going on is that the V5 deletion just makes the cell grow slower, but then the cell cycle itself is not affected directly. So then you ended up with same progression of the cell cycle, but then the growth is less. So then the ratio of the nucleus to cell becomes higher. So summary of the entire talk, by looking at the cell cycle in purulence control and then several cell size related genes. In purulence, we found that there's no obvious connection between cell size and cell cycle. The coordination between budding and cell cycle is also not very obvious. We did confirm that the long G2 in the control or wild type population is due to V1 function, and then bud emergence occurs earlier in the V1 delete mutants, compensating for this shortening of G2. Last, G1 phase is longer in the V1 delete mutants, and then it's not due to the function of V5 gene or protein. So many puzzles remains, but then the tools that I'm currently focusing on is one, what regulates G1S transition, right? So in many of the models that people have looked at, any mutations or DNA damage that induced in G1 is actually repaired in S phase when the replication fork encounters those lesions. So a delay in G1S transition in pool lengths, what may be the molecular player? So second is the question that Danny has been interested in most of his career, is whether to make a bud during a cell cycle, and if so, when to start making a bud and when it stops. So the current working model that we have is probably DNA replication and budding are regulated by two separate regulators. And there is some communication between these two regulators, but not as tight as in Saccharomyces cerevisiae. So besides that, the other thing that I did not talk about is looking at cell cycle control in different morphology. So because different morphology will have different events that need to occur or does not occur. So for example, in hypha growth, there's no cytokinesis, right? In non-bud mitosis, there's no bud need to be made, so there's also no cytokinesis. So how are those components play into the cell cycle control? Influence is another thing that I'm interested in studying. So with that, I would like to acknowledge the wonderful Love Lab. It has been a joy to work in the lab. And then our neighbor, the Bio Lab, has been also very helpful in helping me design my experiments and interpreting my data. So I'm happy to take any question or curiosity you might have.
All right, Yiqiao, thank you for that very interesting talk. Very crazy organism that you have there. What you kind of started to talk a little bit about the question that I had. I mean, as I understand it, Polans is pretty good at growing in all kinds of different environments. Like it's a very common yeast. And so I was wondering are you growing it in like synthetic complete medium with glucose and maybe if you grow it like in a different medium, you'll see, I don't know, the emerging importance of G1 control. So for example, in budding yeast, sort of like the slower you grow it, the relative importance of G1 S control becomes more important. Whereas like if synthetic complete medium in glucose, it's proliferating very quickly. It has kind of a short G1. There's less of an effect of G1 mutations. So I don't know, what kind of media have you grown it in, and are the kind of genetics that you told us about general to all those different growth conditions?
Yes. Unfortunately, I haven't tried. So the other thing is that we have been growing it in YEPD, so the cells are probably very happy in there. But we haven't actually altered nutrient conditions, which is actually what we talked about this morning, to look at what is the first, I don't know, or what is the part of the cell cycle that's most affected when we alter different parameters. But as far as I know, I think someone in the lab have even tried to grow them in water, and they were able to grow in water, which was crazy. So it will be, yes, something that I will be looking into probably in the next few weeks. Yes, in different nutrient conditions.
Okay, thank you. Dave, if you want to unmute and ask your question.
So, there's a very bizarre growth pattern of pool lands. I think you already answered the question that its growth is a lot more prolific in more bizarre conditions than Pombe and Cerevisiae. So now I'd just like to ask, there's this, the previous talk basically took budding as the onset of S phase. And you're showing that in Pulalands, budding can proceed, before S phase is even entered. So, besides cell wall and ribosomes, I suppose, filling out the bud, what do you think is happening during the budding that can be separated from DNA synthesis?
So we don't actually have the answer to that question, but then my thought has always been that I need to prove is that there are two separate networks or pathways to regulate this two process. To be honest, I really don't know. But then originally I thought is the synchrony between these two. Let's say they both oscillate, and then when they are in sync, they bud. When they're out of sync, they don't. But then now I'm looking at the data that I have now that I think that is too simple a model to use. But there are also occasions where the G1 phase is super long. So I can't even quantify in my movie, like 6-hour movie. So then the cells just stay in G1. So those cells I never able to figure out why they did not enter. So the current thinking, at least we have in the lab, is there must be some molecule that is expressed or repressed stochastically besides their regulated weight of expression or translation. But there are something that is stochastically inhibit that those molecules may play a role in the variability in, the entering of S phase.
Do you think it would be worthwhile comparing Pulalands to these more conformist, highly regulated strains like Pombe and Cerevisiae to see, why it's so liberated?
Yeah, that is the curiosity we have in this lab. Like even transition between forms is another curiosity we have. Because obviously they can do it. So right, in some pathogenic fungi, the morphogenesis is just one direction. They think of it as development process. But in Pululans, all these morphologies are actually reversibly. So it's reversible transition. So you can bud from the hypha, so you can go through non-bud from a budding cycle or go through a budding cycle from a non-bud cycle. So we, as of now, have no clue what is going on. But
all right. So next up, we've got Jamie.
Yeah, thanks. So it seemed like the nuclear division cycles were uncoupled from kind of cell growth and the actual cell division cycle. So I was wondering if you looked at nuclear size, like you were measuring cell size and trying to correlate it with some of the transitions, but I wondered if you'd measured the individual nuclei and their size in any way to see if that's correlated with any of these transitions. In particular, I'm thinking about like your Wee1, Wee5 double mutant where you've got multiple nuclei in one cell.
Yes. So I guess there are two parts in my answer. So one is proportionally a smaller size cell, its nuclei will be smaller. But then the ratio between the total volume of the nuclei, of all the nuclei, against the volume of the cell is actually pretty linearly correlated. So that's already in, I think, Allison's paper. But that is more like at the population level, and I think is at some specific time point. But I have not looked at throughout cell cycle.
Okay, thanks.
So next we've got Drish. So Drish, you're in time this time to ask your question.
Yes, thank you. Yeah, I was wondering about the histograms you showed about the the bud emergence onset in wild type and Wee5. And at least in normalized time, they look remarkably similar. At least the means seem to line up quite well. Do you think this is set by kind of a timer, maybe from the start of G1?
So the thing is, they are normalized times, right? But then if you look at the absolute time, it's different. But again, I guess the thing I did not take into account is the growth rate difference. That is actually another. I think Allison is looking at growth rate, but then I guess it's more like delta volume. I guess will be the more appropriate parameter to look at, whether increase in some percentage volume is correlated with the budding. But I don't know. So most of the data we have play around with it in different ways. But then however we slice the data, there's always some inherent variability that we can't quite account for. But yes, thank you for your suggestion. I guess, yeah, that should be something that I'd look into.
Thank you for that.
All right, so next we have Ivan.
So I was wondering if you looked in your data into so-called adder way of controlling cell size, like many bacterial species add on average the same volume regardless of their size at birth, and that's how they maintain their average size. I was wondering if you looked in your data sets into that, because it's a different way of kind of like controlling your size on average.
So I think I just looked at the data this morning. So it does not seem to be a absolute addition of either the absolute volume or proportion of the volume. That I think we did look into that, or maybe another postdoc. She's more interested in growth and then is that segregation of new organelle to the buds. All we have found that have a similarly linear correlation is at cytokinesis, the total volume of the buds is about 45% of the mother cell. And I guess that is the only correlation that we found. But then other than that, I don't think we have actually found, something that predicts when certain cell cycle event happens.
Okay.
Yeah.
All right, and so next we have Matthew. Matthew, if you want to unmute.
Yeah, I wanted to ask you about, you mentioned that in the multinucleated cell, the nuclei will tend to divide kind of synchronously. And something similar happens especially in fission yeast, when you kind of stop cells going through cytokinesis, all the nuclei, they go through multiple synchronous rounds of division. But when that happens, you can also stop it by deleting Wi1 and breaking the mitotic feedback. So I was curious whether you still see the synchrony in the multinucleated cells when you have your cell cycle mutants.
Yes. Also a multi-part answer. So in some of the cell cycle mutants, so the baseline is most of the time they are still synchronous. Whenever it's asynchrony, it tend to be some errors either in mitosis or some other process that we believe is due to DNA damage, because those nuclei that either lag or they couldn't decondense properly, they will end up forming micronucleus in the next cell cycle, or maybe in the current one. So the healthy nuclei, they usually still go through the cell cycle synchronously, but then the unhealthy ones, they just fell off and then they do something else. But yes.
Thank you.
All right, and so with that, I think we've made it to the end of the session. So let's thank Benedikt and Yiqiao for the two really fun talks, and I'll see all of you back here in 2 weeks.